saturated with oxygen, then when it gets to the tissues, it says, hello cells, and it just keeps, I'm gonna stay in my boat and go back and get recycled over and over and over. I'm never gonna get off this ride. So we have this conundrum of requiring a high affinity to completely load our hemoglobin in the lungs and a low affinity when we get to the tissues. Both events need to occur. So the next set of images look a bit boring, but show an important relationship between oxygen and there's several of these on page 60 in your lecture notes, known as an oxygen dissociation graph. Historically, students hate graphs. They're scary and confusing and all of that. But remember, a picture is worth, what, 1,000 words? You can put a lot of information that's more clearly seen on a graph than trying to write it all out. So we'll go through this a few times. This is identified as the y-axis, and this is the x-axis. Okay? And so often the lower line of a graph is time. So over time, these things change. In this case, the lower axis is considered to be PO2. All right, the pressure of oxygen in the blood, in the plasma. And the y-axis is percent hemoglobin saturation. Okay. So here's normal. And when we draw this, we can't get any percent higher than 100, okay? There's no way you can saturate your blood to 100, your hemoglobin to 110%. However, we can have PO2s higher than 100. Normal is about 100 to 104, okay? But I don't want you to think that this is the same as percentage and it can't go any higher. So our typical oxygen dissociation curve looks something like this. It has two distinct regions that reflect those two conditions that needed to environments that hemoglobin needs to function in. All right? So what we're saying is that at a PO2 of 100, we get about 100% saturation. At a PO2 of 50, we get about 40% saturation, on my graph anyway. Over here, PO2 of 50, you're going to get about 70% saturation. So I need to change my graph just a bit. Had too much of a curve in it.
Are you wanting us to memorize these?
No. Just want you to be able to interpret it. All right, so I still don't like that one. Now, what would be wrong if my graph looked like this? It means at a lower PO2, I could get 100% saturation. I could be up at Mount Kilimanjaro or climbing Mount Everest without supplemental oxygen and still be able to saturate my hemoglobin at a PO2 of 40.
It's not getting off the oxygen's not getting off the
Exactly. That would be great in my lungs, and I'd be transporting a lot of oxygen. But if my tissue PO2 is 40, and I have 100% saturation at 45, it's not going to dissociate from my hemoglobin and supply my tissue with oxygen. It's just going to stay on there and never come off. Okay? So we don't want it to look like this. All right? All right, let's take the other extreme then. Let's say that our PO2 looks like this. No matter how high my PO2 is, I can never saturate my hemoglobin. So this is going to say, okay, fine, this is great in the tissues, it comes off so easily, but in the lungs, I can never pick up enough to keep my body alive. It just binds to the hemoglobin and comes right back off again. And so that's the two extremes that had to be solved by one molecule. It had to have a high affinity for hemoglobin in one location and a low affinity for hemoglobin in a different location. All right? So we see that high affinity for hemoglobin up here, identified as a plateau, and that is in the lungs. The advantage of this is that even at a PO2 down to about 75, I still get almost 100% saturation of my hemoglobin. So I can live. I can go up and ski for the day and not be a totally unconscious zombie, okay? Because I have that wide range, that plateau, that even though so on the actual curve here, I don't really start to see a significant drop in percent saturation until I'm at about 75. PO2 of about 75. Okay. So that allows me to, within the world, we have a great range of altitudes and elevations. So that's good. But I don't want it to stick there. So now as I drop to lower values, once I get below 60, I start to see this steep slope where oxygen is coming off of the tissues very rapidly, off of the hemoglobin very rapidly. And that's in my tissues where I need it to come off rapidly. Okay? I have a lower PO2. So with a high PO2, I've got lots of oxygen around there, so that if an oxygen happens to come off of my hemoglobin, there's another one to take its place. Now, why is hemoglobin changing? Here, my PO2 rather, here in the external respiration in the alveoli, I have oxygen constantly being refreshed with every breath I take, and oxygen diffusing into my plasma, elevating my PO2. In the tissues, and we'll just draw this in the same area. So here's a tissue cell. Because of metabolic activity, the cells need oxygen, right? So now the oxygen is going to be leaving the plasma and moving into my cell. What's that going to do to my PO2? So now in my tissues, my PO2 is going to drop as oxygen moves by its concentration gradient into the interstitial fluid and into the cells. So what does that mean on my graph? So moving this way. All right, I'm dropping PO2, and as I do so, it comes off more and more and more and more rapidly. Okay? Remember that little cross I drew for the hemoglobin molecule? Having this saturated increases the affinity. On the reverse, what happens to the affinity when one of them comes off? It drops. So the rest of them come off even more easily, which is what I need to happen to provide oxygen to my tissues. Okay. Before I go further with this, I'm just going to talk about some conditions that change affinity. Is there any questions on how this works? Okay? Because this is the key concept on how hemoglobin, how uniquely hemoglobin fulfills its requirements to both carry and release oxygen. Now, you'll notice that there's several other diagrams, and so depending on the conditions, we talk about the oxygen dissociation curve shifting. Shifts to the right, or it shifts to the left. And usually it's the steep slope that is affected. So what have I said when the oxygen shifts to the right, does it have a higher affinity for oxygen, or does it have a lower?
Lower.
Let's take a look. Here's 50% saturation. With our normal oxygen dissociation curve, it took a PO2 of this value, 50% is about 30 mils of PO2 to have 50% saturation, all right? When my curve shifts to the right, does it take more or less oxygen to keep it 50% saturated? More. Which means that if we're talking about those musical chairs, the kids are sliding off even faster. Maybe that seat, instead of being like this, is, you know, almost straight up. So when the curve shifts to the right, the hemoglobin affinity for oxygen has decreased. What kind of conditions cause that? Many of them actually change the three-dimensional shape of hemoglobin and make it more difficult, not quite sickle cell, but make it more difficult for just normal hemoglobin to interact and for that oxygen to fit into those four proteins or molecules of hemoglobin. One of them is temperature. Well, that makes sense. Temperature makes all the molecules move around more. So it makes sense that with that extra energy and increased random motion of the atoms, all right, that oxygen's not going to sit in there very long. We've got that kid sitting on the chair, and now just instead of making it oily, we're going to shake it, okay? They're just going to slide right off. So temperature. Now let's change the shape of the chair. Instead of making it flat, we're going to take it and bend it this way, okay? So they don't even have a seat. They've got this little ridge to sit on. So things that change the shape are hydrogen ions. Increasing hydrogen ions decreases pH. And that interacts with the polarity of the amino acids, and the protein changes its shape, and the oxygen just doesn't fit as well in the hemoglobin molecule anymore. Comment?
When you were talking about the effect of temperature, so as the curve shifted to the right, are we basically correlating that to an increased amount of oxygen present in the tissues?
What we're saying is when we increase the temperature, we would need to have more oxygen. I'll show it to you in just a second, but there's two different ways of looking at it. So let's come back over here, okay? As we increase the temperature in the environment, the oxygen is going to come off of that hemoglobin more easily, more rapidly, and therefore to keep it on there, we're going to require more oxygen out here to diffuse into here, so that when that comes off, more oxygen around will make sure there's another one there, because it's coming off so fast, much faster. So the decreased affinity means that this is coming off faster, it's not staying there as long. Therefore, to keep the same amount of oxygen bound to that at any given time, we have to keep increase the amount of oxygen that's around to make sure that there's always an oxygen bound there. And that's increased PO2.
Okay, and so that would result in more oxygen actually transferring over to the tissues, diffusing to the tissue?
It depends. It would, with the shift to the right, there's more oxygen in here, but we're just looking at what's bound to the hemoglobin right now.
Oh, okay. I'm
Okay.
sorry.
There would certainly be, yes, the end result is that it comes off more easily, therefore the tissues have more oxygen available to them. Yes.
I got it.
Which is what happens. Exercise increases temperature. Exercise increases hydrogen ions if we're making lactic acid, okay? Elevated levels of carbon dioxide will cause this to happen for two reasons. Carbon dioxide will, increased carbon dioxide will increase the number of hydrogen ions, because it makes carbonic acid. We'll talk about that after break. But carbon dioxide itself binds to hemoglobin, not the iron, but to the protein, and that also changes the shape and makes it more difficult for oxygen and iron to interact. So it increases hydrogen via forming carbonic acid, and it binds to hemoglobin. Okay. There's another molecule. The old name is biphosphoglycerate. The new name is di. It means the same thing. This is listed. All this stuff is listed in your lecture notes on the next page, 62. So on the bullet, the next to the last bullet, 2,3-diphospho or biphosphoglycerate. This is a molecule that the red blood cell makes. It's a byproduct of its own metabolism. And so the more of this DPG that's created, or BPG, it also changes the shape of the hemoglobin. All right, so as DPG increases, it makes it more difficult for oxygen to remain interactive with the iron ion.
What's the DPG again?
It's diphospho, 2,3-biphospho or diphosphoglycerate. It's on page 62 of your lecture notes. Okay? So all of these are conditions that will free up oxygen available for the tissues. Okay? Yes?
So, all those examples, did you say decrease affinity?
Yes. And therefore more oxygen is leaving the hemoglobin and available to go to the tissues. All right, so now if we talk about the other direction, and we move our oxygen dissociation curve to the left, what are we saying? We're increasing. So oxygen and iron are less likely to dissociate from each other. Now that's good for this, we can carry more oxygen, but bad for the tissues, as you'll see, okay? So this is going to result in, where shall I write this? I'm going to write it here, just assume it's going for that, okay? So this is going to result in a shift to the left, reflects an increase in hemoglobin affinity, and the opposite of all the things I wrote up there are going to cause a shift to the left. So instead of an increase in temperature, a decrease in temperature. A decrease in hydrogen ions, which means an elevated pH. A decrease in the amount of carbon dioxide, or a decrease in the amount of DPG. In addition, two other really fun things. Okay, fun just because it's physiology. Carbon monoxide and fetal hemoglobin will also shift the curve to the left. Anybody know why? Where does carbon dioxide binds to the protein of the hemoglobin molecule? Where does carbon monoxide bind to?
Does it bind to the iron?
It binds to the iron. It displaces the oxygen. It has a higher affinity. It competes for the oxygen on the iron. So yes, the hemoglobin can carry less oxygen. All right? But it also, the reason it can compete is because it has a higher affinity. So once it does get to the iron, it stays there longer. It hardly ever comes off. So what does that mean for our tissues? What are the signs, the really characteristic signs of someone who has carbon monoxide poisoning? Now, most people that don't get enough oxygen, they turn pink or blue?
Blue.
Blue. Okay. Cyanotic fingernails are gray, purple. Someone with carbon monoxide poisoning has really pink fingernails, cherry red lips. That doesn't sound like you're not getting enough oxygen. The problem is the oxygen is not coming off of the hemoglobin. So yes, the hemoglobin has less oxygen on it, but remember what happened to affinity? So we bind a carbon monoxide molecule, or maybe two. That actually makes it easier for oxygen to bind because it increases the affinity for oxygen. But guess what? It increases the affinity so much, even if this one is oxygen, that it doesn't come off. So not only is it taking the place of one or two oxygen molecules, but the oxygen that does bind, when it gets to the tissues, notice what happens? The affinity is increased. Carbon monoxide increases the affinity of iron for oxygen, hence the shift to the left. So now, at 50%, our PO2 is much less with the same amount of oxygen saturation. So as the tissue PO2 drops, the oxygen still doesn't come off of the hemoglobin iron, hence the cherry red lips. Okay, and yet the tissues aren't getting their oxygen. So the tissues die because the oxygen is still in the blood. Okay, PO2 is normal because it's still equilibrated. So that oxygen will go to the tissues, the oxygen that was dissolved in the plasma, but the oxygen that's bound to the iron won't come off the iron and take its place. And that's where most of the oxygen is. There's not enough oxygen in the plasma to keep a person alive if this is significant. All right, so that's why carbon monoxide shifts our dissociation curve to the left, because it increases the affinity of hemoglobin for iron. Treatment, hyperbaric chamber, increase oxygen concentration. So even before they get the person, there was over in Woodland, or Winters, I guess it was, there's a nut packing plant, and they had some carbon monoxide poisoning incident quite a few years ago, in the last 10 or 15 years. So at the site, they're going to give them oxygen, trying to increase the amount of oxygen, okay? So that would eventually, if that carbon monoxide molecule ever came off, there would be so many oxygen molecules there that even though they came off more frequently, they would kind of flood out and dilute the carbon monoxide. Same reason for the hyperbaric, but it's just more effective. I mean, there's a limit to how much oxygen you can actually give somebody to breathe. And so the hyperbaric chamber just increases its force and pressure and the likelihood that when a carbon monoxide molecule comes off, there's enough oxygen there to prevent it from coming back and binding to it again. Okay? All right, so that was carbon monoxide. And we'll do fetal hemoglobin, and then we'll take a break and come back and look at carbon dioxide briefly. Okay, so fetal hemoglobin. We've already talked about fetal hemoglobin because once the baby's born, it stops making fetal hemoglobin and has to break down those red blood cells. And we talked about the bilirubin levels going up and jaundice and so on. So normal hemoglobin has 2 beta and 2 alpha chains. All right. Fetal hemoglobin has the alpha chains, but the beta chains are replaced by gamma hemoglobin. Gamma hemoglobin, or the gamma protein part of the hemoglobin, the gamma chains, when they interact with the alpha chains, have less affinity for oxygen. I'm sorry, more. Let me get backwards here. Have a higher affinity for oxygen. This is needed because the baby's lungs are the placental area, and we have less oxygen available there, so we're starting off with a lower PO2. So if we want to be able to transport, if that's the lungs, it really needs to be high PO2, but it can't be, so what we do is we change the affinity. Now, is that a problem with the fetus for tissues? eventually, you know, so if this is the placenta, the fetus, we start here instead of here, all right? But we have a higher affinity for it, so it still works okay, because our whole line is shifted to the left, so it still works. And it still works here because this slope is shifted to the left as well, because the fetus tissues are at a lower PO2. All right, so adult, air-breathing humans, our tissue PO2 is about 40. I don't know what fetal is, but it's less than that, so there's still a gradient. And so the oxygen still comes off of the hemoglobin. A lower PO2 is required for that to happen because of that higher affinity, but that's what the fetus has, is that lower PO2 at the tissues.
So you said adults have 2 alpha, 2 beta chains, what does the fetal have?
2 alpha, 2 gamma. Okay, so they have the higher affinity. There's another interesting thing that goes on. Remember the DPG that I talked about? The mother's red blood cells are making DPG. That increases the amount of oxygen that she's giving off at the placenta. The baby's red blood cells, the fetal red blood cells, are unaffected by DPG. They don't make it, they aren't affected by it. So the DPG in the blood of the mother can move into the baby's blood, it moves across the capillary wall. And at the mother's red blood cells, it decreases the affinity, so more oxygen comes off of the mother's red blood cells. But it doesn't affect the fetal hemoglobin. So we maintain the higher affinity, even with all the mother's DPG around. Okay? So unaffected by DPG. Now, some adults have a disorder in which they make fetal hemoglobin. Okay, yeah.
Is it supposed to be DPG or BPG?
Either the BPG is an older name, but it
Oh,
means the
so
same
it's the
thing,
same
die or buy.
Okay, fine.
So it means the same thing. So let's say that you have a disorder in which instead of making the alpha beta chains, your red blood cells are alpha gamma. So even though you're 22 years old, you have fetal hemoglobin. What does that mean? Where would it help you? Where would it hurt you?
It would help for binding the oxygen.
Okay, so you would do fine, you would do better, it would take less time to acclimate
But it wouldn't come off.
at a
of the
higher
tissues.
altitude,
But it wouldn't come off in the tissues.
but it wouldn't come off the tissues as easy. You would have to have a lower PO2, your tissues would be really starving for oxygen in a relative component to somebody who had DPG, okay? So you'd have to have a lower pH or a warmer temperature of the blood or more CO2 to get the same amount of oxygen to leave the hemoglobin as if you were making DPG. Okay. All right, let's stop there and take a break. I need to stop talking for a minute. And when we come back, we'll spend about 30 minutes 35 minutes talking about carbon dioxide transport. We've got all the basics. Actually, we don't have an carbon dioxide dissociation. We just need to talk about the formation of carbonic acid. So it'll take us about 30-35 minutes, okay? And then we'll be done. Some of you already started the lab on the spirometry last week, Tuesday. And so lab today is going to consist of giving you the opportunity to finish that, and we have the quiz that you guys didn't take on Tuesday. So it's the same quiz that I wrote up for Tuesday. I haven't changed it. I haven't looked at it since then. And so it's the anatomical structures of the nasal cavity, pharynx, cartilages, nerves, muscles, parts of the lungs, but not any histology. So I'm not going to be showing you any histology slides asking you to identify type 1, type 2, respiratory bronchial, bronchus kind of stuff, structure. Okay? All right, so why don't you come back in quarter after, okay?